Catherine, Simone, Henning. This is my third visit here, and it's really, I don't think that all of you students realize how valuable it is for us to get phenomenal refresher on electrophoresis and hear from one of the world's experts on mass spectrometry. You know, you'll hear this whole week the speakers are just outstanding, and we never talk about these nuts and bolts, these kind of basic things that we do. So it's incredibly valuable for all of us also. And the other thing that I always find amazing is when I first got into this field in like 1999, the work that I would have presented at a poster last night compared to every single one of yours would have looked like it was done by a kindergartner in the last century. Just because the field has advanced so much and the capabilities and the biology that people can pursue now is really spectacular. So the bottom line is you're all really lucky to be here. Okay. As am I, for that matter. As am I. So just a few of these slides will be refreshers. Obviously, proteomics plays an incredibly important aspect of biological research. There's certain things that you can do by protein mass spectrometry you can't get any other way. And the correlation between mRNA and protein expression is still an active area of research, and it will always be so because it's very complicated. There's really no other way to get post-translational modifications other than doing protein mass spectrometry, kind of in a, especially for discovery-based approaches. And something that people will get more into, you'll hear from Bruno about quantitation, how important that is. I'll talk a little teeny bit about it. But really, proteomes are incredibly dynamic, and we need to come up with ways to, first of all, capture all the information and also study the dynamics. And that requires really powerful technology, running from the analytical chemistry aspects to things to mass spectrometry, chromatography, and then data analysis. You can obtain peptide information on proteins. Mass spectrometers are sensitive and fast, and they just get, and now you can do proteins to a certain extent, and they just, as you heard from Alexander, they get more sensitive and faster, and more sensitive and faster every year. The poster that I would have presented 15 years ago is on a piece of equipment that probably no one has anymore, called an LCK Classic. And it almost probably would be impossible to get a paper published using that piece of equipment in today's day and age, although it's still near and dear to my heart. Shotgun proteomics is very similar to shotgun sequencing. You know, basically everybody's referred to, Albert and Thierry, is that you have this problem, we have this complex mixture of proteins that we're making even worse by breaking into peptides, that we then have to use really sophisticated chromatographic approach and mass spectrometry approaches to try to interpret and put it all back together in order to capture what was the proteome that we studied. An old slide that I made in a kind of a hand-drawn version of a cell that's really bad that I did years ago is shown up there. But basically, you know, anything that has a protein in it, you can try to purify, isolate, analyze, and put through a process in order to get to this step where you're going to have your identification of proteins. You know, you heard about the sample preparation aspects of it. There were workshops yesterday of different components. Today I'm really going to focus on this LC-LC, or just the LC part of it. You heard about the mass spectrometry, and a little bit out of order because the chromatography comes before the mass spectrometry, but so be it. And I can't possibly talk in the period of time, really for a week, about chromatography and give it sufficient due. It's obviously an incredibly well-established, really long-standing field, and it's become really critically integrated into proteomics, say in the past, I don't know, probably 20, 25 years. There was a gentleman named J.C. Giddings from the University of Utah who published all these theoretical papers on multidimensional separations that I've always thought were brilliant because he was thinking about things that simply couldn't be done at that time. Where really we have these really complicated mixtures, and we'll get more into this, that you have to be able to separate by two or more independent separations. And orthogonality is really critical. Something that's separated in one has to be able to be maintained separated in the other. Oh, there we go, thank you. So you resolve in the first, you have to resolve your entire process. It's basically impossible to really do, but you make an attempt. And the power, this is a typo, this should be peak capacity. Resolving power is measured by the peak capacity, which is the maximum number of peaks that can be separated in a given column. So basically there's about 7 steps that you'd say in a shotgun proteomics pipeline. We'll get into little bits of all this, but mostly we'll be talking about the chromatographic aspect. Obviously there's the generation of peptides. You have to prepare and load, run, acquire your mass spectrometry data, match the peptides. You'll hear a great deal about this from another one of the world's experts, from John Cottrell later. Filter and assemble, do your statistical analysis in order to come up with your list at the end. So, as you've heard from Albert and Thierry in particular, this generating of peptides is a very complicated approach. There's membrane proteins, there's soluble proteins, there's proteins in different parts of the cell. There's different enzymes you can use to digest that you're going to get different results. And really, you're taking this very complicated mixture, and again, you're making it worse. So, as Albert also mentioned, just using trypsin, for example, you're going to cut these tryptic peptides out. But there's a lot of reasons to want to try to use other enzymes to try to get higher sequence coverage, to cover more of your protein in order to do so. And obviously that just makes things even more difficult, especially say if you're going after post-translational modifications. And the pipeline that we worked on a long time ago is to come up with a system where you're using different enzymes to be able to look at different parts of your proteome. And again, these are really complicated mixtures each individually. So how do you go about analyzing and processing all this information? So let's take a really simplistic kind of schematic conceptual aspect to begin with about how, let's just pretend this is a little reverse phase column that has way more than a peak capacity of 8. So this is kind of where this idea of orthogonality comes into play. So probably most of you have done reverse phase chromatography prior to a mass spectrometry to acquire your data. And it can be done over the course of all sorts of different periods of time. And it works really effectively to a certain extent today's day and age because of the power of mass spectrometers, but you're not going to ever capture everything. And I would, in a little bit, I'll do something rather egregious with someone's from this audience's recent paper to actually prove my point. So let's say you have a peak capacity of 8 in this reverse phase column that's about like this long. If you double the length, you're basically going to get a peak capacity of 16. But if you use 2 different separation techniques, that say reverse phase or strong cation exchange, which I'll talk about a great deal today, your peak capacity is now 64, even though effectively you have the same length. So because they're 2 totally different separation techniques, you've got this 8 times 8, 64, so there's a lot more kind of separation power in that system. So, and this is the case for many different types of approaches. For phospholipid work, I'll mention a little bit, you'll hear a great deal more about. People use, say, titanium dioxide or iron IMAC to be able to enrich for phospholipeptides, then followed by reverse phase. Those are 2 orthogonal techniques. You can use a variety of different chromatographic approaches like strong cation exchange, weak cation exchange, strong anion exchange. If you do that type of fractionation prior to the reverse phase, you're going to get in this type of particular case. If all you're doing in reverse phase in a given length, you have far less actual peak capacity or resolving power than you would do if you do something else with half of your distance or half of your steps, basically. Hopefully this is clear. It's a very simplistic representation of a really complicated problem, but it's really important to have these orthogonal separation techniques that basically are separating on different biophysical properties as part of your system, as part of your pipeline to really get good coverage, really no matter what you're doing. My group almost exclusively does protein complexes, and you'd think we'd be doing overkill, but we still focus on doing these types of separations because we want to know as much as possible about a sample in any sample that we analyze. I mean, philosophically, it kind of boils down to the far and away the most valuable resource in all of science is people's time. And so when someone takes the time to prepare a good sample, it's worthwhile trying to know as much as possible about it, given what you're trying to analyze in your biological problem. And as probably the first principle of proteomics is always going to be the case, and it's part of this idea of using powerful techniques to analyze as much as possible, is garbage in, garbage out, that was stated a couple days ago. You take the time to do a good sample, take the time to do good chromatography, take the time to do good mass spectrometry. Otherwise, you're basically wasting your own time. So this is not that old, but reasonably, about 10 years ago now, a paper from analytical chemistry where they're talking about orthogonality. So you can kind of envision, here would be two different separation phases, but basically you can pretend that this would be like, say, 10 centimeters of reverse phase and 10 centimeters of reverse phase. You're not separating, so you'd have 20 total centimeters. There's no orthogonality, so everything's just going to be eluting along this line. And things are going to get compacted, so you have peptides basically eluting and overlapping. If you had perfect separation, which is effectively impossible to achieve, each one would be completely isolated in space and time at an individual moment. As far as I know, there's no technique, chromatography technique that'll do this, and there probably never will be. Unless you use really, really simplistic mixtures, I suppose it's possible, but I don't know of anybody who's been able to prove a claim along those lines. What usually typically happens is something like this. So if you have, say, a strong cation exchange or reverse phase, you get reasonable separation, but if this were the millions of peptides that really typically come out of a digestion, it would look a lot more complicated. But you're getting things in places where you can start to simplify your mixtures in periods of time so the mass spectrometer can acquire the data. You saw a little bit about peak resolutions also the other day. It's also kind of important to think about where peptides are typically identified. So as chromatography is going along and a peptide elutes into a mass spectrometer, you don't typically get Gaussian peaks, or as Alexander referred to, the St. Louis arch look like a chromatographic peak, which is probably the first time I've ever heard such a crazy thing. Really, you'll get a peak that'll go like this, and it'll have a tendency to tail off. Most mass spectrometers are set up where they're going to have this kind of line in the sand drawn, where below a certain intensity threshold, you're not going to acquire information, and once it goes above it, it triggers an event to acquire information. And so typically what happens is as the peptides are eluting along, they start to accrete, you're going to have this, you might be sampling here, but typically you're usually going to sample here as the peptide elutes into the mass spectrometer and goes above a certain level. So it kind of be in between this front cutting or heart cutting. Usually you don't sample peaks after they've started to tail off, but that's not universally the case. But what you're trying to do is you're trying to elute a peptide in the mass spectrometer so that it gets captured somewhere during this point to give you sufficient signal, sufficient intensity, so you can get good tandem mass spectra or get good signal from your mass spectrometer, whatever it happens to be you're trying to do. All right, that is a really fast kind of theoretical summary of peptide chromatography to a certain extent, and now we'll go into the details of the different types of methods that are used in proteomics. So, chromatography is a massive field. Far and away the most commonly used is reverse phase chromatography, which you're separating on hydrophobicity. There's strong and weak cation exchange chromatography, which is going to be SCX or WCX. If you were to go to Phenomenex, Waters, GE, Thermo Fisher, and look at these catalogs of different chromatographic materials that are available, there's a vast array, even within each one of these categories, different types of things. And there's other additional phases too that we don't use a great deal. There's normal phase chromatography, which is kind of a variant of this thing called HILIC that people use to look at, say, really hydrophobic peptides. I don't have time to really go into that too much detail. You'll hear probably more from Judith and others perhaps about phosphopeptide specific with this iron immobilized metallophytic chromatography or titanium dioxide based chromatography. I'm going to spend a few minutes, not very much, because you've heard quite a bit about phosphoproteomics, about how this particular approaches work too. And it also is a good example of incredible complexity and why you need to do quite a bit of chromatography at times to really capture it all if you really want to be comprehensive. So here's a really simplistic picture of reverse phase, where we have basically our silica with a long, very hydrophobic chain sticking off of it. Lots of different types of moieties can be put on here to kind of vary it, but this is the basic, and it's obviously not that big. These are very small particles that get used, but effectively you're separating a hydrophobicity. Strong cation exchanges are very simplistic. You're going to have that same kind of silica bead, but on the end of it you might have, this would be a benzene sulfonic acid, where at very low pH you'll be able to have pH 2, you're going to be able to have peptides bind this because the pK of this is even lower than that. Anion exchange is just the opposite, where your stationary phase is positively charged, so you have to have your peptides in a mostly negatively charged base state, so you could be altering your pHs to be able to use this. Cation exchange is going to be negatively charged, so you're going to have your peptides in positively charged state to be able to bind to the peptides, to bind to the particles. And one of the reasons why strong cation exchange has become so prevalent for coupling with reverse phase chromatography for proteomics is because you want to do things at low pH. At low pHs, peptides that are positively charged, and that's how we're basically all running our mass spectrometers in positive ion mode, so that you're looking at a tryptic peptide that has its lysine on the tail that's positively charged. It helps it then fragment. So one reason strong cation exchange has become really well adopted is because you're at the right pH basically to be able to do things, and it can be directly coupled to reverse phase as part of a pipeline. All right. And in terms of the technical aspects of how chromatography works, that's really about it. Basically, you have a lot of different types of methods that are out there. The predominant one is reverse phase. There are others, including weak anion exchange, strong anion exchange, but in those types of cases, prior to getting it under a mass spectrometer, you're going to have to change your buffer system, basically get it to low pH to couple it to reverse phase. Strong cation exchange, because you're already at low pH, in terms of the buffer that you're using, is more directly compatible to mass spectrometry. But there's a lot of other approaches that you can use. So, prior to me going into excruciating detail about mud pit, we'll do a little bit more about why I use more than one dimension for separating peptides. And I'm going to use somebody else's paper, a recent one, to help prove my point, so you just don't have to believe me. Obviously, reverse phase, as I mentioned, is most commonly used. Probably everybody in this room has this. If you have a system that's been set up and you haven't really looked at it that closely, maybe you have, that's what you've got. You've got a reverse phase column, there might be a trap, and then there's an analytical column going directly into a mass spectrometer. It's a one-dimensional separation technique, and it can't possibly capture everything. And you really need to use multiple dimensions, sometimes 2, 3 dimensions, to really get good depth of a proteome. So, very briefly on phospholipeptides, a lot of really spectacular work has been done on phospholipeptides, and my group doesn't do much of it. We follow others when we try to. So, this immobilized metal affinity chromatography, this iron IMAC, was probably, I don't know, the Hunt lab was involved early on in development of this, but a lot of people have done a lot of nice work in this area. But one of the first big kind of proteomics papers in phospholipeptides was a Nature Biotech paper a bunch of years ago from Don Hunt's group, where they used iron IMAC. So basically you have iron that's going to be coupled to beads, and you set your conditions up properly so that your phosphate groups are going to be bound to your IMAC, and then you can use different types of approaches to elute them off. There's also metal, I don't remember what the O stands for, but basically you can use titanium dioxide in a similar type of manner on beads, so that it's attracted and binded to phospholipeptides that you can then elute. There was a poster last night about using gallium. There's a lot of different metals that you can use in, hopefully Judah will cover this in more detail, but there's a lot of different metals you can use in kind of in a bound manner on beads to bind phospholipeptides and elute. Also with phospholipeptides, there are phosphotyrosine antibodies people use to IP phosphoproteins or phospholipeptides to analyze. This mostly works for phosphoserine and phosphothreonine, and not as well for phosphotyrosine. Would be the general consensus, but not universally true. Also, say for example, Judith with Steve at Harvard, and when Steve at the Gigi lab started looking at strong cation exchange at a particular point in time, they noticed that phospholipeptides were eluting at a particular point in time. So you can also kind of use phospholipeptides. They'll elute really early on in strong cation exchange. And so if you focus in on this right kind of moment in time, you can also capture phospholipeptides quite nicely. And a lot of work has been done over the years where I was guilty of it too for a long time, It's like my method is better than your method. This is a very, very common title of probably, of this, it should be the title of a lot of proteomics papers, because you develop, first people started doing this iron IMAC, and then, okay, now we're going to do titanium dioxide, and it gives you a little bit better results. And now, well, if we do strong cation exchange and look at phospholipeptides, we get better results, and it goes back and forth and back and forth and back and forth. So method development works, is you have to compare what you've done to what's the prior knowledge and be able to make claims. Part of the reason for all of these, well, my method's a little bit better than your method, or is better than your method, is because no one has really, I think, spent sufficient number of time thinking about how to capture them all. And I won't be able to do justice to this paper, and there's a couple people here who can probably explain a lot better than I can. I'm guessing, is there someone whose last name Ruprecht in the audience? That person's not here. Yeah? Is that you? Raise your hand if this is your publication. No? All right. Is anybody here on this paper rather than the actual author? The people at the back don't matter. We have the corresponding author, and I don't know what Bernard probably did. Anyways, the point of this proves this concept of, so this was, what were they, A431 cells, if I recall correctly, human tissue culture cell line, that basically you're chopping up into peptides, using this IMAC approach to isolate all your phospholipeptides. And the typical pipeline would be here. So, even though you're reducing the complexity of your mixture by just looking at phospholipeptides, you're still tons of them in the actual cell. So the typical pipeline would be, people will do their iron IMAC, their titanium dioxide, perhaps gallium, and then go down this pipeline. And yeah, 4,000 to 5,000 phospholipeptides is cool and a lot. But then someone does maybe the exact same cells and a little bit different, and they're going to get, if they were to do a Venn diagram, they would overlap to a certain extent, and people would say, well, these are complementary methods. Instead, somebody decided to do a great deal of work, and using strong anion exchange, because you're dealing with negatively charged peptides here, so you want to have a positively charged molecule to be able to get them to bind, generated 24 fractions. Now, this isn't directly compatible with reverse phase, so you have to do some buffer exchange to get into the system. But so, and then you have 48 hours of LCM SMS, and now you've basically gone on to 10,000 to 15,000 phospholipeptides. So you're getting the depth, exact same samples with replicas, properly done, and really only because you're adding this extra separation step, you're capturing what's really present. Evidence of absence isn't absence of evidence, right? So here, you're just not sampling sufficiently to capture what all is probably there. And it's possible that this isn't even sufficient, I don't know, but really I think in me cursory following the phosphorylation field for a number of years and not doing really much research in this area, this is the first time I think I've seen a paper where I thought maybe they're getting close. And this is the type of work that you really need to do if you want comprehensive work. You got to do the extra work. The sacrifice is obviously time. Here you do it in 4 hours, here you do it in 48 hours. So, but the question is what's the value of that time? You went through this process of doing all this work, you designed your experiment, you grew your cells up, maybe you're doing a drug treatment or whatever. Take the time to do it the right way. Take the time on the mass spectrometer, take the time to do the chromatography to get this depth. Because once you have this information, that's much more biologically rich, obviously, than here. Especially if you start doing, say, quantitative analysis of phosphorylation networks. Have all of you read this? I just cut this directly out. So often quoted complementarity of different materials for phospholipeptide can actually be attributed to limited binding capacity, biased or incomplete elution, shortcomings in the physical formats, and limited analytical capacity for mass spectrometry. So really, to a certain extent, that sentence is like all of you other people who have been doing this for a long time have done a crappy job. It's basically what the two of them just said, but in a nice kind of way. And really, the point is, though, is they did sufficient chromatography to capture all this information. All right, I've beaten this horse to death, but I think it's a really good example of, well, no matter what you're looking at, it's worthwhile thinking about more than one fractionation step. You can't just do reverse phase for 4 hours into a luminose or lumos or whatever it's called and get everything. There's too much complexity in the proteome. Okay, so there's a lot of different ways to couple dimensions, first dimensions and second dimensions, into a mass spectrometer. There's a variety, you heard about two-dimensional gel electrophoresis. Thierry also talked about the gel-C MSMS approach, where you run a gel, cut it into bands, digest all the peptides, all the proteins in a particular spot on a gel, and then run those as fractions into a reverse phase chromatography. There's capillary isotopic focusing, I won't talk about it all. Capillary zone electrophoresis. There's a wide variety of different approaches. Most things are going to be coupled to a reverse phase right before the mass spectrometer. It's just an incredibly well-established, very effective approach. You can directly electrospray things off of a reverse phase column into a mass spectrometer, and that's huge for all of us. Now, what I'll spend the majority of the time talking about here is strong kinetic exchange and reverse phase because they're directly compatible. One issue is always going to be if you are doing offline fractionation, like say for example in the phospholipeptide paper I just briefly covered, it's a lot of work to collect all those fractions and then buffer exchange them to go into the reverse phase system. So you probably are using an autosampler and sampling it out. So you have this other time dimension of your sample preparation. These can be more directly coupled, and we'll go through some different types of approaches that are out there. I always like this technique a little bit too, this IPGIF reverse phase of peptides rather than proteins, but I don't think it's used very much. Waters has developed a high pH, low pH reverse phase tandem mass spectrometry approach I'll spend a couple slides on. That's also an orthogonal approach because you're basically, it doesn't quite satisfy the theoretical aspects of orthogonality, but you're doing it at two different pHs, and so reverse phase behaves very different at high pH and low pH, so it works quite well. I know people who have switched from this to this in the past few years. And I won't go into it very much, but the gel-C MSMS approach, where again you run a single SDS page gel, cut your bands out, go into a reverse phase tandem mass spectrometer. This is a very common technique, and that is a protein separation followed by peptide separation. It works quite well for a lot of people. I don't think it's the type of thing that you want to use if you want to get really deep into a tissue or a cell. It's probably most suitable for, say, doing protein complexes because your mixture is a lot less complex than, say, looking even at the mitochondria or the nucleus. If you want to do those types of things like tissue or organelles or whole cell lysates, you need to think about these other types of approaches. So, for strong kinetic exchange and reverse phase systems, there's a little bit of history. Basically, mass spectrometry companies like to sell, those of us who are lucky enough to buy things, entire systems, where here's your HPLC with all of these connecting tubes and all these things that can done. Here's your mass spectrometer. It's fully integrated. All you got to do is stick something in the autosampler, and data will magically come out of the end. And I don't think that that's ever really happened and been successful. Part of it is because it becomes very complicated to do some of these types of things, and you get these dead volumes. So dead volumes are really important when you separated a peptide out and you have this nice peak. If you then have this empty tube where there's just basically HPLC fluid flowing through it, the bands are going to start to flatten out. You're going to get band broadening. And that's then going to make, and unless you are able to reconcentrate that later, your signal intensity is going to drop down, and it might go below this threshold where the mass spectrometer won't acquire it. In the thermal instruments, it's called automatic gain control. You set this number above which you trigger an event, below which you don't. So if your bands broaden too much in these dead spaces that can occur as you go from the strong kinetic exchange to the C18 trap and then the analytical column, your band broadening will cause you to have decreased numbers of indications at the end. And that happens in all of these types of systems that are kind of fully integrated. So here you have this, you're putting it onto your strong kinetic exchange and you're eluting in a bump type manner onto your trap because in this case you might be using sodium chloride or potassium chloride. Next you're going to wash to do this by changing your valving system. And then you're going to have at the very end, you're trying to elute by going from a C18 trap onto a reverse phase column into a mass spectrometer. Now, the strong kinetic exchange and reverse phase systems, this is one that, an example of something that basically everyone basically had at one point in time. Waters had one, Thermo Fisher had one, Phenomics had one, trying to do this. And I would be able to take, and I had one of these, and I could do the mud pit I'll explain, and compare it directly to this, and mud pit would blow this out of the water using the exact same amount of material because of what's happening right here. You're doing this washing step on the C18 trap, and then you're eluting it onto this reverse phase column. And now you can do some tricks and have some different reverse phase materials here, but basically, you're eluting off of here, it's going to band broaden this entire way, and it's just going to basically wash right through to a certain extent. Because it's coming out here, hopefully it's getting reconcentrated and done again, but it's almost like you're taking a reverse phase and putting this little chunk in the middle. And it's not a very effective technique to try to do a strong cation exchange reverse phase approach. And this is the entire pipeline. That, frankly, I don't know how many people actually do this anymore, if I haven't in a long time. The mud pit approach I'll talk about a great deal more is a direct approach where we're basically packing these little these few silica columns with the material directly so that we can run an inefficient form of strong cation exchange, but much better than doing it in this column switching based approach into a mass spectrometer. You can also do things offline. So this comes down to a time component, though, and this was a common one for people to do, although they still took this type of approach sometimes, or you could do like a fast type approach to filter exchange, to change things out, to go directly in here off of an auto sampler. But the typical way of doing chromatography has always been is you have your HPLC, your FPLC, you do a chromatography, collect your fractions, and then work your way into it. This is the paper from Bernard and Simone on phospholipeptides. They basically took this type of an approach to a certain extent, doing strong anion exchange. After doing that iron IMAC or titanium dioxide IMAC, you would then collect weak anion exchange fractions prior to going into this reverse phase type of system. I don't know if your guys' system has a trap prior to it or not, but this would be a typical kind of offline approach where you collected all your fractions. Now this takes time, obviously, and then more time to get into here. So there's this gap that will also occur. And then again, you're going to have these steps that have to occur. Some of the systems have this reverse phase trap into the reverse phase. And I just don't know if anybody's come up with an approach where this really truly functions as well as it should because of this dead volume. At one point in time, many years ago, this friend of mine who's now at Columbia, he had taken one of these things and then filled all of these things with, so it was like one giant reverse phase column. It kind of worked, but the back pressures were enormous, and it was basically a pain in the ass. But it worked better than this particular approach by itself. So we'll come back to that a little bit more. One approach also that can be used, given the existing equipment. A lot of people have 2D gel electrophoresis equipment in their laboratory, especially this isoelectric focusing, these IPG strips that you can do. And from a group published many years ago, and I show this here every time because I think it's a clever technique, but I don't see it used very often anymore. But basically, you can focus peptides, cut your strip after you've focused your peptides, do your extraction, and then do a reverse phase tandem mass spectrometry approach to look for things. So rather than doing protein isoelectric focusing, you're doing peptide. You do your digestion, you do your peptides, cut it out, and that's a second, a different dimension step for separating out peptides that you can use to expand your depth of coverage. They did this in a comparison, again this was many years ago, of an offline strong case to reverse phase. Now these numbers don't look very impressive in today's day and age because it was done, these were done on LCQs, these weren't done on Orbitraps or Q-exactives or fusions or things along those lines, or Q-TOFs or 5600s, but here they were saying that they were orthogonal and complementary techniques, but really is probably the case where at this particular point in time, we still weren't getting the type of depth that was needed to be able to look at whole proteomes, even if I'm fairly confident that it's still the case that it takes quite a bit to get in true depth. But it's providing access to potentially a different part of the proteome that they didn't cover in their particular offline approach. The other one is using this high-low pH. So the water's acuity system, this nano acuity system that they have, is an ultra-high pressure system that they've coupled directly into their time-of-flight instrumentation or their Synapse instruments that you can purchase. One of the things that they do for getting high resolution, for really good chromatographic approaches, is this 2D HPLC using high and low pH. So you'll have two reverse phase columns. Yeah, I have that in the next slide to a certain extent. It's two reverse phase columns where you, first one, you're doing it at pH 10, and then you do some buffer exchange on your second column, and you're doing it at pH 2, which again, here you're at the proper pH to be able to go into the mass spectrometer, your positively charged peptides, and can acquire information. So there's just some, here you're doing ammonium formate at a high pH, you drop everything down, and you're doing reverse phase gradient. And that, I think, has gained some traction in the field. Again, it's somewhat of a somewhat complicated type of system where your reverse phase column here is after your first valve. This is where you're going to do your high pH work. It's got to come onto here for your trap. So here they're using two different types of C18 to try to deal with this potential band broadening issue in order to try to reconcentrate prior to going to the mass spectrometer. And again, I know people that have switched from strong cation exchange reverse phase to this reverse phase system. It doesn't necessarily have to be purchased from Waters. You can design and set this type of thing up on probably almost, you probably can't, you need a quaternary pump. So you need a pump, you need an HPLC pump that has at least, well, probably 3 channels if I'm thinking about this correctly. Can't use a binary pump, you need 2 binary pumps to be able to set this up with 2 separate valves, in a laboratory. Or you had 1 quaternary pump, meaning you have 4 different buffer channels that you can use to be able to do this type of thing. It's something that we've kind of always wanted to try and directly compare to the mud pit approach that we use, but it just hasn't, we haven't gotten around to doing it yet. Yeah, and here's a picture of the exact one that they have on this nano acuity that I took directly from a Waters poster off of their website that obviously I forgot to give their attribution to, but effectively, it's very similar, although I think they changed this particular one over the years in terms of how you're going to go about doing this particular approach, where you have a rather complicated pumping system to get these different buffers into it and eventually do your elution into the mass spectrometer. So they have 2 different pumps they're using for sample injection and fractionation. So one over here and then over here. If anybody's ever seen it, it's a pretty big box. And there's lots of secrets inside of it, which is probably one of the limitations. So they then, and again, this first kind of came out from Waters over a decade ago now, where they were looking at a lot of different types of approaches in terms of trying to figure out what their peak capacity was, and which I mentioned earlier is a really critical component for the higher peak capacity, the more resolving power, the greater depth you can get of your proteome. And the way that they calculated it out is their peak capacity of this high-low reverse phase was more than double of their particular 2DLC strong cation exchange. Also, the strong cation exchange Hillick approach, or Hillick C18, they found was closer to this high-low reverse phase. And then, say, doing size exclusion chromatography, and C18 had the lowest peak capacity. So they were basically trying to make an attempt to say that this had the highest potential peak capacity also. And that may actually be true. I don't really know exactly how people can precisely calculate this information. You can kind of theoretically determine it, but I don't know if there's really a practical method, like they're calling it practical peak capacity. Because if you get more than 5,000 peptides, that means that something was probably not quite right in your calculation. You always get way more than 5,000 peptides when you do an analysis. But I haven't followed the chromatography field closely enough to see if someone's come up with a good way of actually calculating it directly from any given system. Say after you've done an experiment, how would you calculate capacity? I don't know the answer to that, but it would be really useful to be able to do so, because then you can really directly better compare, and you can analyze all of your experiments in I think, a really effective manner. But this high-low pH, I think, has gained some good traction. This was an offline version that they set up on their own system. So I think this type of an approach doesn't perform as well as a mud pit-based approach. Hillock C18 is compelling. I didn't talk about Hillock very much, but it's a variant of normal phase chromatography, which is like the opposite of reverse phase. If you want more information about Hillock, you can feel free to talk to me. Has anybody in this group ever used, have you used Hillock? Anybody used Hillock? I mean, it's pretty effective. There we go, I have one hand in the back. So everybody here, talk to her about using Hillock and how effective it is. But it's a pretty effective approach, especially say for really hydrophobic proteins. I'm sorry, for hydrophobic peptides. But it satisfies quite a few of this orthogonality criteria. All right, so I have spent all the time I'm going to spend really talking about other people's stuff and other techniques. Now we're going to go into probably nauseating detail for some of you guys about mud pit. But I think the thing to keep in mind is don't think about this so much as a strong cation exchange reverse phase particular approach. Think about, you guys are all the new generation. Think about other ways that you could combine Hillock reverse phase, high-low reverse phase, it can't be done here, or maybe an anion exchange or some other chromatographic approach. Think of it as kind of an analogy for a way that you can think about doing things if you have the right types of systems around. And even though it's kind of sad, we've developed this type of an approach a bunch of years ago. When I was with John Yates and Dirk Wolters, there was a whole lot of us who were involved in this. Here's an old picture of an LCQ where we have this, and that is actually tape, to tape down the column to go into the mass spectrometer, but you can use this for anything, for cells, tissues, proteins. My group in particular, if I have time, I'll spend a little bit about what we use it for, but just a few slides. We really are focused on protein complexes, especially in chromatin remodeling and transcription via our own research and via our collaborators, is really what we use this for. Again, you're taking your protein mixture, making it worse, more complicated. It's more complicated, but you're also to a certain extent, you're making it somewhat more homogeneous mixture a little bit because you're breaking everything up in these particularly sized pieces from much more kind of variant. And instead of having things in lots of different states, you linearize them. They have positive charges across them by dropping into low pHs. We have these things, these pressurization vessels where we do all of our work, our packing and loading of columns to go into the mass spectrometer, and then you hear an enormous amount from one of the world's experts in, I guess, probably about an hour and a half from John. On this particular step, I'll go into just a little bit about our pipeline at the end, because everything you've heard about is just one piece of this overall pipeline about how to do proteomics analysis. So this will be some of the really practical aspects of things. Who here has a laser puller in their group? So I have one. Is your hand really up? Or that's the royal we elbow up wrist? You have a laser puller. Do you use it? Do you pull your own columns?
Yes.
Yeah? And where is it that you're located? Do you work for someone in this room?
It's in Copenhagen University.
Okay, that's cool. No one else has access to a laser puller? We got a couple more. Where are you guys located?
University of Southern Denmark. So it's.
Oh, and are you with, who's your?
Yeah, I'm with Frank Kelsen.
Okay, all right, cool. So you guys have the ability to, one, save an enormous amount of money over the time, because purchasing these columns, in the United States, there's this company called New Objective that sells a strong cation exchange reverse phase column. It's just 2 phases. It costs about $500. When we make one, the total cost of our materials, and this is about a $12,000 piece of equipment. We have these made by a machine shop in Kansas City, Missouri. And when I first sent them the schematics to make these things, they were like, we can't make that thing for you because, if you don't properly fix this thing, this metal piece is going to go flying off into the ceiling and kill somebody. And I'm thinking to myself, well, yeah, if you're stupid and sitting there like this, looking over it, unscrew it. And then, so some lawyers had to get involved, because we had these in the Yates lab. Some lawyers had to get involved, and finally it was decided, as long as you put this danger high pressure sticker on all of them, you can do whatever you want. So there you go. And we used to call them bombs, pressure, mud pit column bombs or whatever, and obviously that's not cool. So our high pressure vessels, where, clearly it's dangerous. Although all right, sorry, I know I'm going on the side. This postdoc of mine named Amber Mosley, who now she's an unbelievably incredible person, and has an incredible story about where she's from and how far she's gone. She has her own lab at the Indiana University Biochemistry Department of the Medical School. And she once managed somehow, I've never seen this happen before, where she was packing a column with this fused silica and it blew up. And she had her glasses on, but it got all these little pieces of glass embedded in her finger. And at first she thought that she'd lost her finger because it like desensitized. And all these things happen, there's all this paperwork, and she goes off to the clinic that she has to go to. And the doctor's sitting there thinking, like, what did you just do? Had absolutely no idea how to do any of this, take care of any of this thing, because had never seen fused silica embedded in somebody's finger before. And then, so she just got released, went home, and her husband just got some tweezers, and they just plucked all the pieces out, wrapped it with the Band-Aid, and she was fine. That's the only injury that's ever happened in my group with one of these types of systems. And it was actually because the fused silica itself, we buy these long rolls. At the very end, when you get to the really bottom of it, if it's been sitting for a long time, it'll be fragile and break really easily. So that's something that comes up pretty commonly, where we'll have columns break when we kind of get to the end of, say, if we bought 100 meters of this stuff. We typically now buy it in smaller chunks. Anyways, so you have fused silica, which is 350 micron outer diameter, 100 micron inner diameter is the common one that we use. We have our laser puller where we can pull these columns and do lots of crazy things. Our different packing materials. Here's a Wattman Partisphere strong head exchange. This is a Phenomenex Aqua. I don't think we use these two anymore because they don't make them anymore, but there's different names of the same thing. First, we're going to pull our column. We're going to pack it, put reverse phase, strong head exchange, reverse phase. We'll wash it to buffer exchange and load our samples inside of these pressurization vessels. So here's a picture of fused silica that was taken many years ago, a couple different HPLC columns. Obviously, this was taken at the University of Washington, where I started my postdoc. You have a piece of fused silica that you've basically cut off a long piece. Inside your laser puller, you're going to pull it. And we used to look a lot at the tips after they were done, but we don't anymore, because it basically works all the time. You're going to first load with your packing material. So inside of this pressurization vessel, you're going to have a little Eppendorf tube with some HPLC material and some methanol. Stick it in, cap it, and pack it. You're going to wash it, open up this pressurization vessel, put it in a separate Eppendorf tube with the strong head exchange material, pack it, close it off again, and then we'll put the reverse phase material at the end. Now we do this for online desalting. So when we've gone through and purified our protein complex at the very end, you're in a buffer that's compatible with trypsin, you're at 2 molar urea, it's a tris buffer. Lots of people, we used to do solid phase extraction, basically prior to loading onto, we would just have a 2-dimensional column. So we do solid phase extraction, and then to get it into our buffer system, you can use a fast type of approach to get into your buffer system to be able to load directly on there. But then when we started doing this online desalting and just taking our digested material and loading it directly onto here, we're sort of getting better results, although I'm sure we're getting some sample loss, and things are just washing through, because this doesn't have nearly the binding capacity that strong head exchange does. And again, inside of here, we have our non-desalted sample. So, obviously getting your peptide mixture into the right kind of buffer environment, low acetonitrile, water, and then formic acid. In our particular case, people use different types of acids to be able to make it compatible with the mass spectrometer. We used to do this 2-phase approach where we do offline desalting. It took longer and didn't give us as good of results as doing this online desalting. Now, one thing to keep in mind about this, if you take this type of an approach, it's not 100 meters, sorry. It's 100 what does it even mean anyway? anyways. 100 micron inner diameter fused silica. Not too sure why that happened. This will be at very high pressure and take a long time to load if you have a lot of material. So if we have higher amounts of material, we can take a different 250 micron inner diameter fused silica and pack it and use this a fritted-based system that you get from Upchurch to be able to load a lot faster, and then we can put an analytical column on the end. This type of split three-phase system we use a great deal, especially when we're looking at a project with someone at the Stars Institute studies planaria regeneration. Planaria are these crazy worms that you can cut up into pieces and they fully regrow. It's a really interesting kind of model for stem cell regeneration. And they want to do whole proteome analysis. And we get a lot of material in a decent sized volume that's impractical to do here. It takes forever to load. It'll take overnight to load. So we use this type of an approach a great deal when we say have a milliliter of material that we want to be able to load onto a column. We'll use this, flow it into waste as it's loading. There's always some sample loss, but that's the case with everything. And then we'll put this analytical column on prior to us running out of the sample. So here's a better picture of our dangerous high pressure system where here's an actual, and this is literally a picture from the laboratory, where we have Kim White that's collecting things as it's falling off, and you can see it, you can see the material coming down. And behind all of it, we always have, in this particular case, this is the wall that's been painted back, black. There's also another room where we have a whole bunch of these things where there's just shades pulled down so that you can actually see the material loading into the column. It's one of the challenges of doing this type of thing, is to be able to watch it. To try to be able to reproducibly put the same amount of material every time on there. It's a little bit of an art, but it's been reduced to practice. So the idea of one of these pressurization vessels is pretty straightforward. You got your peptides at the bottom, air's coming in, pressurizing it, and hopefully, mostly things are binding to this reverse phase. And if I go back, these kind of little dark circles, those are going to be basically gigantic salt molecules, if you want to talk about it literally. So peptides are binding, some are being lost probably, some are binding to the beginning of the strong cation exchange hopefully, but all the salt's being washed off. So we can buffer exchange this so that now this is ready to go directly under the mass spectrometer. And literally they come from these pressurization vessels. You pull the column off, place it directly in line with the tandem mass spectrometer and a quaternary HPLC pump. By quaternary, I mean it has 4 buffer channels, where you have low acetonitrile, high acetonitrile. And to do this online, strong cation exchange works best with some sort of sodium chloride, potassium chloride as your eluate. You have to have that sodium or potassium molecule to push things off. That will cause a giant crust of salt to form on the entrance of your mass spectrometer. You have to use semi-volatile salt, either ammonium acetate, which is what we use, or you could use ammonium formate. It causes quite a bit less salt buildup, if any at all, but it will, at high concentrations, sometimes the spray will die during certain points of the experiment, but I'll get to that. So we have it placed in line between these two systems. Your loaded column is going directly on here. Obviously this is a schematic. First step is reverse phase gradient, where what we're doing is we're moving peptides off of here onto the strong cation exchange, and some things are going to probably go onto the strong cation exchange. And then you have your salt pulse to go on to the beginning of the reverse phase gradient, and a reverse phase gradient to go to the mass spectrometer. I'll give a better picture of this in a little bit. And then you're going to do this by increasing your salt pump. So it's not a really classic strong cation exchange approach where you're eluting things off and collecting fractions. We're doing pulses of salt, reverse phase gradients, higher salt, reverse phase gradient. And the idea is you're trying to move certain portions of your peptides from the strong cation exchange to the reverse phase, and then run your reverse phase into the mass spectrometer. The reason for this is that you're trying to give the mass spectrometer time. You know, the whole premise of all of this chromatography is we can't take a peptide mixture and give it to a mass spectrometer and get everything. Mass spectrometers take time to be able to, you know, as Alexander showed yesterday, things go into the Orbi trap, they get analyzed there, part of it goes into the ion trap, they get fragmented there. There's this time component. So you're trying to introduce as few components to the mass spectrometer at any given point in time so that it can acquire information on it. And that's the idea that's going on here, is you have your salt bump to move some portion, your reverse phase gradient into the mass spectrometer. So here's a little bit more modern picture of an LTQ, which is actually my favorite instrument of all time and remains so this day. It's like having, it's like the difference between an old Ford Thunderbird that's all mechanical and has no electrical parts that you can fix yourself and take care of, versus these Lumos, which is like a Lamborghini with tons of electronics that costs a fortune, that looks awesome and scares the crap out of you when you're driving from Monaco to Genoa and it comes out of your rearview mirror. That happened to me a couple days ago. But when they break, it costs a fortune and no one can figure it out except people in Italy. Anyways, and we use these really old HPLC pumps, these Agilent systems that have just worked really well for us, and they're very inexpensive. So again, here's our system coming in from the pump, our columns. A little bit better schematic of our first step, this gradient of acetonitrile. Oops. So again, up here, we're moving our peptides to the strong cation exchange. Hopefully this is mostly what's happening. The next step is a series of salt concentrations where your first salt bump, some fractions moving to the reverse phase. Then you have this wash point to get rid of the salt, and you do your reverse phase gradient to try to introduce a few components into the mass spectrometer at any given point in time. Next, as you go to, actually you need the higher acetonitrile ones to get the more hydrophobic peptides off. Those hydrophobic peptides will probably never come off this reverse phase column, or we see them very, very late in our analysis because we do this massive blowout type of step of super high acetonitrile. That's where techniques like Hillick become really valuable, I think. Next is going to be a higher salt bump, different peptides from strong cation to reverse phase. You have your wash, acetonitrile, elution to do a few peptides at a particular point in time. And so, just again, the main technique that people are going to be using is just from here to here, right? Just a reverse phase gradient. And this shows it. So you don't have to think about all of this as just in terms of mud pit, strong cation reverse phase. This is also what happens is if you're doing a reverse phase column, you have your peptides bound here. If we go back, basically at this step, you're doing your acetonitrile of some sort of different type of gradient to try to introduce a few peptides in the mass spectrometer at any given point in time. So let's take just a little bit of a look at, what was this one? This is here is a HEK293 cell analysis of this something called the halo tag, affinity purified Borel A protein, which is from the NF-kappa B protein interaction network that we've been studying. But it's just a data set. And this is something I think people forget also. So you do all this chromatography. The mass spectrometer is also like another dimension of isolation and separation. It itself, because they're so fast and so sensitive, is separating things out and choosing at particular points in time. If it can sample your chromatographic peak faster than things elute, you can get really good coverage, which is what happens. So let's look at this. So here's one particular mud pit step that it shows, and here we're at this particular time point, 55.41 minutes in, and here's this mass spectrometer, acquired over a fraction of the period of time, and basically you have your peptides that have eluted. At that particular moment in time, we're dealing with the, where is it? 814.34. That's the MS1 that's been chosen for collision-induced association shown here. So it's this particular peak and the next one that's been isolated. Now we've been fragmented. This information is what you're going to use for your database analysis using Sequest, Mascot, Andromeda. This is what you're using to do your actual analysis of your peptides. Here's your experimental tandem mass spectra. The next one is 742.23, which is somewhere in this particular area if we were to blow it up. This isn't a particularly good tandem mass spectra. There's probably some sort of neutral loss that's occurring. I have no idea what these particular peptides in this one are. We're just doing to see how things are working. Here we did this 1249.48. This is probably a +3 peptide. So that dude was somewhere buried down here. This is how this kind of dynamic exclusion works on these systems, is you choose a high peak and you work your way down. And we can go through of doing 5 MSMS in this particular case, and then another another MS scan. So the whole point of all this chromatography, any chromatography you do, is to separate out peptides to be able to pull this off in any mass spectrometer. And as mass spectrometers get faster and faster and faster, this becomes more and more feasible to do, to be able to sample across all these peaks. And this isn't a really complicated one right here, sorry. It doesn't look like there's a lot of peptides eluting, but there really are a great deal of them. And there are other points in time, like if I were to go into here or over here, any of these ones, there's lots of information that's present that you want to try to sample. So that's the whole point of this chromatography, is to try to get your mixture as less and less and less complicated once it goes into the mass spectrometer, so it can do these types of things. And this is the case for any mass spectrometer, even though this is off of an LTQ, a thermal system. But any mass spectrometer we're trying to accomplish, as soon as you go off that column into your mass spectrometer, you want as few components as possible for it to deal with, to get this type of tandem mass spectrum. Easily interpretable, not controversial, peaks will be matched really well, versus say, that was not so good here. I guarantee you that there's tandem mass spectra like this in everybody's publications that we can't do by hand per se, but a computational approach can do. But that's not nearly as high quality as, say, this. And this is really what the goal should be, getting high quality tandem mass spectra data. So again, we're at, so here, we're now at 55.44 seconds. This is the runtime RT, right? So we've gone from 55.41 seconds to acquire an MS scan. We've got 5 MSMS. Now we're at 55.44 seconds, so it's 0.03 seconds basically have transpired to go from MS scan to 5 MSMS to one more MS scan. And this happens over the course of 24 hours, right? So you're basically getting hundreds of thousands of MS files that you need to be able to interpret. You hear about from John in much more detail. But I wanted to put this in perspective too, that all of these systems are coupled together. Okay. So, you've heard a great deal about strong cation exchange in reverse phase. I mentioned briefly this isotope focusing IPG of peptides in reverse phase coupled systems. There's high-low pH systems that I talked a little bit about that's pretty common, a system from waters, this nano acuity they do this with. I didn't go into the gel CMS approach, reverse phase. Thierry covered that a little bit. Does people basically understand what this is? Or want me to draw a picture over here? Clear? This is clear enough? Does anybody use this particular approach? Anybody ever run a silver stain? A one-dimensional gel, cut out bands, digested it, run in a reverse phase. No one's ever done that? All right, so maybe you don't know. Well, good then. Everybody's doing mud pit? Is that what you're telling me? No, I didn't think so. What do most people use? People mostly just doing reverse phase, tandem mass spectrometry? Anybody want to volunteer and answer? Tell me. I'm going to pick on someone. Who's done a proteomics experiment in this room? Start with that. Anyone? Come on. Has anybody done anything other than just reverse phase? Okay, go ahead. Tell me again. What's your pipeline?
High pH fractionation.
You've done the high pH, low pH. On the water system?
Yes, yeah, that's right.
Is it the nano acuity that you've been using? Or it's done?
I didn't do it myself yet. I'm going to, so I'm not so accustomed to this.
What's the mass spectrometer it's coupled to?
That's a Q exactive agent.
Okay. So it's Q exacted. Is it a Thermo Fisher pump on the front end of it?
Yes, it is, but the high pH fractionation was done offline.
Okay. So you're doing offline high pH, and then you're going into the autosampler
Exactly.
before you do. So there's probably a trap.
No.
No trapping column. So how are you doing the buffer exchange?
As far as I know, we do a purification of the samples before, and then you just load them directly.
So basically, there's a sample processing step in between your high pH fractionation and before it can go into the analytical column, into the mass spectrometer. So it's a reverse phase, reverse phase. Good. It's 2 dimensions. Anybody else? So you do the IPG, IEF on peptides?
Yes,
So you've digested up your sample onto peptides.
I run with then I digest them, and then I do fractionation.
Do you cut it up into strips?
Yes, from the strip, yeah,
Right.
I cut it, and then I do mass spec on it.
How many pieces of,
22 fractions.
I'm sorry?
I get 22 fractions.
22 fractions. And then I'm presuming you're going into an autosampler?
Yes.
And is there a trapping column on that system? Or do you go directly to analytical column?
We have a pre-column, analytical pre-column, and then it goes to the reverse phase.
Okay. All right, so there we go. We've got 2 people in here who are using 2-dimensional chromatography. Obviously, I know some group in here has effectively used iron, IMAC, weekend exchange, and reverse stitch. So there's 3 dimensions that we use there to get the 15 million phosphopeptides. Anybody just using reverse phase to do? No one knows. My only point would be, and I guess it's probably fine that you didn't raise your hand because I'd be shaming you. No matter what you're looking at, you need to think about using more than 1 dimension of separations. High-low pH, isoelectric focusing of peptides prior to reverse phase. These are very effective techniques. And if you want to spend all of your time and career comparing and contrasting them all, great, but it's probably a waste of your time. All of these approaches work. What's closest to you is usually the best thing to use, what your collaborators are best at, but think about doing more than 1 dimension. So I did this a little bit out of order. I was going to ask questions after this one. Which shotgun approaches should you use? Depends on what tools you have available, what mass spectrometers, what HPLC, what expertise is available. So here we have a case where you haven't done the reverse phase yourself yet, but it exists in your laboratory. Another case where the IAF reverse phase tandem mass spectra is being done. That's awesome. You're the first person. I've come here 3 times, and no one has ever volunteered that they do this. So I'm glad to hear that actually it's being adopted. And I'm glad to hear this is being adopted too. Although I'm sad that more people don't adopt this. That's just because I'm selfish. But in my opinion, a big advantage of this is there's no fraction collection and no trapping column. So keep in mind, in any of these systems that you use, you want to minimize the distance of once you've separated out something before it gets onto the analytical column, because band broadening will cause your signal to decrease and potentially give you fewer identifications that you can get. So if you have these really long pieces of fused silica where it's just buffer and sample going through, you want to try to minimize those types of things. It's not possible to basically bring it down to 0 unless you have a system quite like this. It's just something that you should keep in mind. So I'm going to do a little bit more about the few additional steps just as a setup kind of for the rest of the week, I suppose, and for this afternoon. Here's a picture of one of our LTQs looking out across the street in Kansas City, actually getting some data done. All of you guys know this tandem mass spectra works by, in a collision cell of some sort, breaking a peptide, a smiley face, positively charged peptide, into this latter series of ions. You need, really, genomic databases are critical. I know there was someone yesterday in the quantitations did a poster who was working on, where's the P guy? Someone was working on Ps and doesn't have a genome database yet. It makes it very difficult to do and challenging, so more power to you. There was someone else who did a poster on snake venom, and that's, I think he said that there's going to be, the king cobra genome is going to be sequenced and finished soon. So that's a really challenging problem that those of us who are lazy stick with organisms that have good FASTA databases. You can get your identifications, your sequence identification is, this is still an ongoing area of research, but really it's basically taken care of. You can do PTM analysis, you're matching, John will talk about this a great deal more, matching your spectra to peptides. You get back in the day when I first started, I could actually do this, take a really good tandem mass spectra and do it by hand. John Yates used to make us do that before we were allowed to start doing experiments. Fortunately, he doesn't do that anymore because you can't do it anymore. It has to be done computationally. We use Sequest and something called Prolucid a lot. You'll hear more about, great deal more about from John. But really the key thing is having a good computational environment. We have a very powerful cluster and data storage to deal with all of this, to do these interpretations, to get your peptides. But you still have to then in silico recombine everything to get to the point where you're getting your lists of proteins and peptides to kind of bring it all back together. And that's really the beginning of your whole process, is having this long list of proteins and peptides. You might have quantitative information you want to use also to be able to do things. We have an in-house kind of software tool where we're able to basically get our peptides. This is going to be spectral counts shown here, sequence coverage, and then we use this quantitative approach called the normalized spectral abundance factor that I won't talk about really at all. But one thing that you can kind of see here, and part of the reason I show this is here is, what was this one? I don't remember exactly what this data set was. It's some transcription. We've got RugB1, RugB2, is a flag affinity purification. I don't remember exactly what it was. But anyways, here's our spectral counts. So it was probably a flag affinity purification of the tau binding protein. This would be, so this is TIP49 actually. Okay, I know what this is. We got 5,000 spectral counts is what we're using in this particular case. This is what MudPit does. This is why we can use spectral counting really only for MudPit. You'll hear from quantitation about spectral counting, and it's probably the most poorly used technique because people aren't using the right techniques to use it appropriately. You can use it when you get a lot of them, but you can't use it if you're just doing a 2 hours reverse phase gradient into your mass spectrometer. I reject those papers when I get them to review. You need a lot of them, which is what you get from an approach like MudPit. But you have to be able to process your information and put it all together as part of what I'm showing here. And then you got to do statistical analysis. And I put this in yesterday because of a conversation that we had yesterday, just briefly. T-test is completely inappropriate to use on proteomics data of any type because it's this many genes, few replicants problem. T-tests are only appropriate when you have thousands of measurements of the exact same thing. So for spectral counting based approaches, we've developed something called PearlJam because Norman was a huge fan of PearlJam. It's from Italy. It's really called the Power Law Global Error Model. He developed it for Affymetrix data that we then adopted. And the only really important point I want to make here is, so here's a plot of intensities of these spectral counting based values. So low abundance to high abundance. And here's a plot of the ratio. This is stationary phase to log phase in yeast. It's completely inappropriate statistically to draw a line in the sand and say anything above twofold, I'm going to say, is overexpressed or significant and below. You have to do statistical analysis. And the way these approaches work, here's just signal to noise of any given individual point, the replicates. So sometimes by chance you get really good signal to noise, and this would be like like a t-test type of an approach. But really what effective techniques do is they sample variance across the entire way and model error. This is what our approach does, and then there's something called Q-spec from Alexei Fischke does. So at very low abundance, you need to be much more conservative, really high fold changes, because there's so much variance in these low abundance changes. These things are just not statistically significant. And at higher abundances, you can get better, lower and lower variances because your signal intensity is higher to get statistical analysis. This is simply just a point I wanted to make for those of you who were here yesterday is you need to think carefully about how you're doing your quantitative analysis. You'll hear more about quantitation in much greater detail from Bruno later that'll cover quite a bit more and in much more depth. But basically, this is also part of our process. And I know I went through this last part really quickly because you're going to hear about more of it later, but I wanted to put all of this into perspective. Everything you hear this week is part of the step of doing an entire experiment. You got to prepare your sample, generate your peptides. We just talked about columns, running columns, really the chromatography today. You'll hear about tandem mass spectra, data processing. You'll hear about quantitative analysis and statistical comparisons also later in the week. I think I have 5 slides left because I just want to talk about a little bit about, to put this in perspective of what we do and what a lot of my colleagues who use this approach. What do we use this for? So, it's a biological discovery tool, and we're getting hundreds to thousands to tens of thousands of proteins. You can look at anything. And really, as I said earlier, the most valuable resource in science is people's time. And what we use, the reason we do all mud pit all the time is we want to know as much about the sample as possible. It's the reason to use reverse phase all the time. It's the reason to use IAF of peptides followed by a reverse phase for everything. You want to do within reason, as comprehensive analysis as you can, because you've taken the time to design these experiments and purify your samples or isolate your samples. We're really focused on interaction networks where we affinity purify proteins, analyze by mud pit. We use label-free approaches. We don't just use spectral counting anymore. We're also using area to the curve and ion intensity, something that we've developed in the past year, where we're looking at networks. We can look at modularity of complexes, which we use by doing deletion analysis, and you can do relative abundance of protein complexes. So this is just to show really briefly the types of things we're interested in and what we use MudPit for. I actually think MudPit is best suited for protein complex analysis more than any other real technique. It's worked very well. It gives us very good depth and breadth of protein complexes. And really our main research right now is to look at how their protein complexes networks are organized and regulated. Rather than just building bigger and bigger pictures and having bigger and bigger hairballs, we want to try to figure out mechanistically what's really going on inside of these complexes. We use some crazy state-of-the-art data analysis techniques, one called topological data analysis from Stanford and the company. And really what we do a lot of is dissecting protein complexes with different molecular biology or genetic type tools. And this is what we use for all of it. We really are very narrow-minded to a certain extent and use MudPit for everything. The strong cation exchange, reverse phase, and the data analysis pipeline is what we use exclusively on every instrument. We have 7 LTQs, a Q Exactive, an Orbitrap Velos, and 2 Orbitrap Elites. And it's got to be 99.9% of the time, it's MudPit analysis being done on them. So, the very, very end of all of this. My objective was to cover something that's uncoverable in a time, but it's just to try to give you guys a little bit of a flavor of the different types of things that are being used. There's a lot of literature out there about chromatography and proteomics that you can look at. Instrumentation manufacturers and especially the chromatography companies have really good kind of, Phenomenex has this really wonderful, just a PDF on their website of peptide and protein separation by a bunch of different things. Waters has a pretty good website. I stole some of the slides from them. Thermo, GE, Healthcare, they all have this information about these different chromatographic techniques. PolyLC, Andy Alpert, probably has the best website for really getting information about HILIC. There's quite a bit of information in the literature too, but this PolyLC is probably, it would be a great place to go and read about HILIC if you want to learn more about it. Obviously, reverse phase chromatography is the most widely used. It is almost always the last step right before the mass spectrometer because you're doing it at low pH. Your peptides are positively charged, which is you're running mass spectrometers in positive ion mode. That's how it works. You need these other steps to be able to get better separation. I should probably also include the isotope focusing on this peptides is also an important additional step to be able to use. I spent a little bit talking about phosphopeptides and used an example from Bernard and Simone's work about how important it is to do these multi-dimensional separations to really get good depth if that's what you want to accomplish. I know it was a little egregious, but I was looking for something and I was like, hey, they just published that. Wouldn't it be fun to talk about their own work? And they can correct me if I was wrong. Here's a picture of the Stowers Institute in Kansas City, Missouri. My laboratory is right here. It's far away. Someone actually from here told me they'd been to this really like world-class barbecue joint in Kansas City, and to say that I was shocked when he told me this is an understatement. Because no one goes there. People fly over it. We'll look up at some point in time and you guys will be flying from New York to San Francisco. That's about as close as most people will get. This is my wife, Laurence Florent. She and I co-run the Proteomics group. We're both we were both trained by the Yates Lab. And then we have a very diverse group of people who run the gamut from computational, analytical chemistry, biochemistry, structural biology, and really kind of high-end sort of systems biology, mathematics, and have a lot of really spectacular collaborators at the Stowers Institute also, even though it is a small, to say it's an ivory tower is an understatement. It's really like a gilded cage because we're very well funded. We're very fortunate to be there. But with that, I will stop. Thank you guys for your attention, and for those of you that I've pestered to answer a couple of questions. And I'll be happy to try to answer any other questions. I'll be here until Thursday afternoon if you want to talk more about chromatography, which is always one of my favorite topics, even though it gets lost in the mysticism of these massive, fancy mass spectrometers. So thank you all for your attention this morning.